Dear friends, in earlier two modules we had discussed about the DNA sequencing techniques. In this third module we are going to discuss second generation sequencing method. After studying this module, you shall be able to understand about the second generation sequencing technologies, and also you would be able to learn the principle underlying the next generation sequencing method and differentiate between the common type of next generation sequencing, namely Polony sequencing and pyrosequencing, and also understand the potential application of next generation sequencing techniques. As regards the introduction of this topic, following the conclusion of the first human genome sequence in the previous decade, demand for rapid and cost effective sequencing methods has increased. This has led to the development of second generation sequencing techniques, commonly called next generation sequencing. Current next generation sequencing platform perform massively parallel sequencing, in which millions of DNA fragments from one sample are sequenced at a same time. These massive parallel sequencing technologies facilitate high throughput sequencing and allow sequencing of an entire genome within a single day. Next generation sequencing is not Sanger based sequencing, but offer high throughput DNA sequencing. Millions of DNA strands are sequenced in parallel, yielding significantly higher throughput and minimizing the need for fragment cloning method that are required for Sanger sequencing genomes. Large scale sequencing is often aimed at sequencing of long DNA pieces, for example, entire chromosome, even though the same can also be used to generate very large number of short sequences, such as found in phage display. For longer target DNA such as chromosomes, a common approach is to digest the DNA with restriction endonuclease or shearing with mechanical forces, resulting in shorter DNA fragments. The fragmented DNA can be cloned in DNA vector and amplified in bacterial host such as E. coli. The short DNA fragments are then purified from individual bacterial colonies and sequenced in isolation, which are then assembled and overlapped into one long continuous sequence. A wide variety of next generation sequencing methods are available. Some of these sequencing methods will be discussed in this module. One of the method of next generation sequencing is shotgun sequencing. The shotgun sequencing, also called shotgun cloning, is a technique used for sequencing long DNA strands. As the name suggests, sequencing is analogous with the rapidly expanding quasi-random firing pattern of a shotgun. Principle of shotgun sequencing is based on the fact that Sanger's method of DNA sequencing can only be used for short sequences from 100 to 1000 base pair, and long DNA sequences must be subdivided into smaller fragments and sequenced and later reassembled to yield overall sequence. Two principal ways commonly employed for this are chromosome walking, which progresses through the entire strand piece by piece, and shotgun sequencing, which is relatively faster but more complex and utilize random DNA fragments. Multiple overlapping sequences are obtained for target DNA by several rounds of this fragmentation and sequencing, which are then aligned using computer algorithms and overlapping contigs, generating a continuous sequence. Shotgun DNA sequencing was one of the prime methods that was used for whole human genome sequencing. To understand this further, you may look on the following two rounds of shotgun reads. Upon sequencing, neither of the sequences cover full length of the original sequence, but all 4 reads can be aligned and overlapped into original sequence using their overlapping ends. In real life practice, the amount of data generated is enormous and is prone to ambiguities and error. The situation is further worsened due to presence of multiple repetitive sequences in the genome. Another technique is Polony sequencing, which was developed by G.M. Church in Harvard Medical School. The technique is highly accurate, multiplex sequencing method, yet inexpensive, and is capable of reading millions of immobilized DNA sequences simultaneously in parallel. Furthermore, the technique does not require specialized hardware and can be easily set up with a commonly available epifluorescence microscopy or a flow cytometer with computer controlled flow cell or fluidics system. In addition, Polony sequencing technology is an open platform with freely downloadable open source software, supported protocols. Sequencing is performed using paired and tags library, in which each molecule of DNA template is of 135 base pair in length with 2 17 to 80 base pair genomic tags separated and flanked by common sequences. The current read length of this technique is 26 bases per amplicon and 13 bases per tag, leaving a gap of 4 to 5 bases in each tag. Polony sequencing is among the first next generation sequencing system that was used to sequence an entire E. coli genome in an accuracy of 99.999% with reduced cost of about 1/19 that of Sanger sequencing. The method of Polony sequencing consists of 3 main steps, namely: number 1, paired and tag library construction, number 2, template amplification, and next is DNA sequencing. As regards pair and tag library construction, genomic DNA is sheared into smaller fragments, which are then subjected to end repair and A-tail treatment. This repair treatment converts any damaged or incompatible sticky ends of DNA to 5' phosphorylated and blunt ended DNA, enabling immediate blunt end ligation, while the A-tailing treatment adds an A to the 3' end of the sheared DNA. DNA fragments corresponding to 1 kb are selected by using 6% TBE page gel. In the next step, DNA molecules are circularized with T30 T-tailed 30 base pair long synthetic oligonucleotides, which contain 2 onward facing MME one restriction site. Following the amplification, circularized DNA molecule are cleaved with MME1, releasing the T30 fragment flanked by 17 to 18 base pair tags, approximately 70 base pair in length. The paired tag molecule need to be end repaired prior to the ligation of emulsion PCR with primer oligonucleotides to their both ends. The resulting 135 base pair library molecules are size selected and nick translated. The resultant DNA contains a 44 base pair FDV sequence, a 17 to 18 base pair proximal tag, the T30 sequence and a 17 to 18 base pair distal tag and a 25 base pair RDV sequence. Next step is template amplification. This step is further divided into multiple sub steps. First of these is emulsion based PCR, in which paramagnetic streptavidin beads of same size are loaded with dual biotin forward primer. Because of strong affinity between biotin and streptavidin, the forward primer will bind to bead surface tightly. Following this, an aqueous phase is prepared with preloaded beads, PCR mixture, forward and reverse primer and the paired end tag library, and is mixed with an oil phase and vortexed vigorously to create an emulsion. Each droplet of water in oil emulsion has one bead and one molecule of template DNA, permitting millions of non-interacting amplification. Following amplification, emulsion is broken using isopropanol and detergent containing buffer. Beads with amplified DNA have less density in comparison and are separated by centrifugation. Beads are then capped at 3' end and attached to the surface of a cover slip. The cover slips are then bonded to the flow cell body. The next step is DNA sequencing. The principle of Polony sequencing is based on the biochemical differences in properties of ligases and polymerases. A series of anchor primers are hybridized to synthetic oligonucleotide sequences at the immediate 3' or 5' end of the 17 to 18 base pair proximal or distal genomic DNA tags, which is followed with ligation of anchor primer with fluorescent labeled monomers. Monomers anneal with differential success to the tag sequences and are selectively ligated with the attached anchor primer and give fluorescence that indicates the base, whether an A or C or G or T occupies the position on the genomic DNA tag. After 4 color imaging, the anchor primer or monomer complexes are stripped off and a new cycle is begun by replacing the anchor primer. A new mixture of fluorescently tagged monomers is introduced, for which the query position is shifted one base further into genomic DNA tag. The cycle is repeated over and over again. Several bases in 5' to 3' direction and 6 bases from the 3' end can be queried in this manner, resulting in a read length of 26 bases per run, 13 bases from each of the paired tags, with a 4 base to 5 base gap in middle of each tag. Figure shows various steps of Polony sequencing technology. Now, we come to pyrosequencing. A parallelized version of pyrosequencing was developed by 454 Life Sciences. The method amplifies DNA in aqueous phase droplets within an oil solution by emulsion PCR, with each droplet containing a single DNA template attached to a single primer coated bead that then forms a clonal colony. The sequencing machine contains many picoliter volume wells, each containing a single bead and sequencing enzyme. Pyrosequencing uses luciferase to generate light for detection of the individual nucleotide added to the nascent DNA, and the combined data are used to generate sequence readouts. This technology provides intermediate read length and price per base compared to Sanger sequencing on one end and Solexa and Solid on other end. In 2008, 454 sequencing launched the GS-FLX titanium series reagents for use on genome sequencer FLX instrument with the ability to sequence 400 to 600 million base pair per run with 400 to 500 base pair read length. Pyrosequencing has several steps. The first is DNA library preparation and EMPCR. Genomic DNA is fractioned into smaller fragments 300 to 800 base pairs and sticky and are converted to blunt ends. Short adapters are then ligated, which provide priming sequences for both amplification and sequencing of the sample library fragments. One of the adapter, that is adapter B, contains a 5' biotin tag for immobilization of DNA library onto streptavidin coated beads. The single stranded DNA library is immobilized onto beads. The beads containing a library fragment carry a single stranded template DNA molecule. The bead bound DNA library is then emulsified with the amplification reagent in a water in oil mixture. Each bead is captured within its own microreactor where PCR amplification occurs. This results in bead immobilized clonally amplified DNA fragments. Now, as regards sequencing, single stranded template DNA library beads are added to the DNA polymerase and are layered with enzyme beads containing sulfurylase and luciferase onto pico-titer plate device. The device is centrifuged to deposit the bead into the wells. The layer of enzyme beads ensures that the DNA beads remain positioned in the well during the sequencing reaction. The process is optimized to contain a single amplified library bead. The loaded picoliter plate is placed into genome sequencer FLX instrument. The fluidics subsystem delivers sequencing reagents containing buffer and nucleotide across the well of the plate. The 4 DNA nucleotide are added sequentially in a fixed order across the pico-titer plate device during the sequencing run. During the nucleotide flow, millions of copies of DNA bound to each of the bead are sequenced in parallel. When a nucleotide complementary to the template strand is added into the well, the polymerase extends the existing DNA strand by adding nucleotides. Addition of one or more nucleotides generate a light signal that is recorded by the CCD camera in the instrument. This technique is based on sequencing by synthesis and is called pyrosequencing. The single strength is proportional to the number of nucleotide, but is linear only up to 8 consecutive nucleotide, after which signal falls off rapidly. Figure explains the comparison of pyrosequencing and Polony sequencing. Other than shotgun sequencing, pyrosequencing and Polony sequencing, there are other sequencing methods like Illumina Solexa sequencing, solid sequencing, ion torrent semiconductor sequencing, DNA nanoball sequencing, heliscope single molecule sequencing, single molecule real time, in short called SMRT sequencing, and massive parallel signature sequencing, MPSS. All these DNA sequencing techniques were developed in order to make the sequencing fast and cost effective. These next generation sequencing methods and program have reduced the cost of sequencing by 6-fold as compared to the classical method of sequencing like Sanger and Gilbert. As regard application of next generation sequencing, it has potential of offering endless applications, allowing for rapid advances in life sciences. To mention a few, number 1 is re-sequencing of human genome is currently being performed so as to identify the gene as well as regulatory sequences and elements. This has further been extended to identify pathological condition. Next generation sequencing can impart knowledge about the genome in comparative genomics of various related species. Next generation sequencing has been applied in field of public health, epidemiology and medical treatment to identify the sequencing of bacteria and viruses, which can facilitate identification of virulent species. It has also been used as a substitute to more technically challenging RNA microarrays by using next generation of sequencing of RNA. This has enabled scientists to identify the RNA sequences that are expressed at any given time point. In summary, we can say that demand for cheaper and rapid sequencing technique have given rise to the development of next generation DNA sequencing techniques. These are rapid, cost effective and perform with high level of accuracy. Multiple platforms are developed for next generation sequencing such as Polony sequencing, pyrosequencing, Illumina sequencing, Solexa, mass spectrometer based sequencing and more. Polony and pyrosequencing are based on emulsion PCR, where amplification of unique sequences is performed following sequencing. Data generated is enormous and is aligned using software. Next generation sequencing has numerous applications in biological sciences, medical and allied branches, and is becoming increasingly popular. Thank you.